In this part of the report, I would be discussing the citric acid cycle. The citric acid cycle is a series of chemical reactions used by all aerobic organisms to generate energy through the oxidation of acetate, derived from carbohydrates, fats, and proteins into carbon dioxide. The citric acid cycle is amphibolic, meaning that it plays a role in both catabolism and anabolism. The citric acid cycle has two other common names. One is the Krebs cycle, after Sir Hans Krebs, who first investigated the pathway. In this work, he received a Nobel Prize in 1953. The other name is the tricarboxylic acid cycle, or TCA cycle, from the fact that some of the molecules involved are acids with 3 carboxylic groups. In eukaryotes, the citric acid cycle takes place in the matrix of the mitochondria. However, in prokaryotes, these steps both take place in the cytoplasm. The 8 steps of the citric acid cycle are the series of redox, dehydration, hydration, and decarboxylation reactions. For step 1, the red type traces the fate of the 2 carbon atoms that enter the cycle via acetyl coenzyme A. The blue type indicates the 2 carbons that exit the cycle as CO2 in steps 3 and 4. As we can see, the red type goes only through step 5 because the succinate molecule is symmetrical. The 2 ends cannot be distinguished from one another. Notice that the carbon atoms that enter the cycle from acetyl coenzyme A do not leave the cycle in the same turn. They remain in the cycle, occupying a different location in the molecules on the next turn, after another acetyl group is added. Therefore, the oxaloacetate regenerated at step 8 is made up of different carbon atoms each time. Each turn releases 2 carbon dioxide molecules and produces 3 NADH, 1 FADH2, and 1 ATP or GTP. The citric acid cycle goes around twice for each molecule of glucose that enters cellular respiration because there are 2 pyruvates and thus 2 acetyl coenzyme A's made.